All right, so just going to launch right into this. We're going to continue on basically where I was in lecture 9 and then get into what we were supposed to cover on lecture 10. Tried to reduce the total amount that I did in lecture 10 in section A, so we're trying to get everything in sync again here. So the basic picture from this analysis of chloroplast evolution is that there are 3 different evolutionary histories represented in the cells that contain chloroplasts. The chloroplast has an evolutionary history grouping with cyanobacteria. The mitochondria have an evolutionary history grouping with proteobacteria. And the nuclear DNA has an evolutionary history grouping in the same general way that for the tree that we talked about and the groupings that we talked about in the previous lectures with those 7 major lineages of eukaryotes. However, the model doesn't fit as well as the model for mitochondrial evolution in terms of looking at the total data of eukaryotes and where chloroplasts are found and what the phylogenetic trees look like for particular parts of chloroplast and eukaryotic evolution. And I'm just going to give you a couple of examples of this before talking about the revised model for the evolution of chloroplasts. So one possibility is that the chloroplast symbiosis happened in the branch leading up to the common ancestor of all eukaryotes, maybe after the mitochondrial symbiosis, possibly even before, but this model would suggest, if this was true, that all of the lineages of chloroplasts, of eukaryotes that descended from this common ancestor at one point in their history had a chloroplast, as well as had a mitochondrion. And one reason to be that people were a bit skeptical of this possibility is that if you look at the distribution of chloroplasts across eukaryotes, this is a tree from version 8 actually of the book, but it basically shows the major lineages of eukaryotes. It has 5 major distinct groups. The exact details for this tree don't matter. The point is to look over here and see the green labels. Those are the species with chloroplasts. Those are labeled green if at least one member of that entire group, like the apicomplexans, has a chloroplast-like organelle. It's so much more scattered than the mitochondrial presence and absence. And even if you look within these groups, many of the representatives within these groups do not have chloroplasts. So if you look at foraminiferans, not all of them have it. Euglenids, not all of them have it, and so on. And so the distribution pattern itself is sort of highly dispersed, highly sparse. And if you hypothesize that the chloroplast was present in the common ancestor of all these lineages, you would have to propose dozens, if not hundreds, of loss events where different lineages that now currently do not have a chloroplast have lost the chloroplast in their history. And even if you proposed this and tried to test it, there's other data that doesn't support this ancestral presence of the chloroplast either. So the proposing hundreds of loss events is already seems a little strange, maybe not parsimonious. But another part of the biological information that led to people to propose an alternative, that is to not propose that the chloroplast was present in the common ancestor of all eukaryotes, is looking inside the cells of various eukaryotes that have chloroplasts. Some of them, those cells look like this, with a chloroplast and a mitochondrion and a nucleus and simple membrane structures consistent with there having been a primary symbiosis of chloroplasts. Primarily the organisms that look like that are in the Plantae lineage. We'll come back to that in a minute. But many other lineages, if you look inside the cell, there's just incredibly strange things going inside these cells that's hard to jam chloroplasts in the ancestor of all eukaryotes into this explanation. So for example, in an organism called cryptomonads, you can see multiple nuclei. One of the nuclei is in a membrane-bound compartment where the chloroplast is also contained. That chloroplast is wrapped up in many membranes, and then the chloroplast and the nucleus are surrounded by yet more membranes. And then There's the regular nucleus for this cell, which has its normal membrane structure. This observation of way too many membranes surrounding chloroplasts and the presence of additional nuclei inside some cells led scientists to propose a modification of the chloroplast evolution model. And the modification goes like this. The proposal is that the symbiosis happened in the lineage leading up to the common ancestor of the Plantae group. If you look inside the cells of members of the Plantae group, they all look basically like this in some way. Simple, few membranes, one or two things around the chloroplast, maybe a cell wall, simple mitochondrial structure, simple nucleus structure. And the proposal to explain, I should turn off the video recording. After this symbiosis happened, then the different lineages within the Plantae group each evolved their own particular adaptations of their chloroplasts. Those accumulated mutations and changes by the process of divergence. And you have what I've drawn in color, these sort of different, here are the glaucophytes, here's the red algae, here are the chlorophytes, here are the land plants. I've just colored them to represent that they are distinct branches, but they all inherited the chloroplast, mitochondria, and nucleus from the common ancestor of the Plantae group. And the proposal to explain this weird distribution pattern of chloroplasts outside the Plantae group is that those lineages that have chloroplasts brought inside of them a symbiont that came from the Plantae group. That is, they had a symbiosis not with the chloroplast itself, but with a eukaryote that contained the chloroplast. I'm going to show you the model, sort of the cell engulfment model to explain this. So we had some normal eukaryote with just a nucleus and a mitochondrion, and that engaged in some type of symbiotic interaction with a representative of the Plantae lineage, probably a single-celled representative of the Plantae lineage. It engulfed it, brought it inside the cell. That's an endosymbiosis, just like the endosymbiosis for bacteria and chloroplast origins, but now it's a eukaryotic cell being brought inside. This is known as a secondary symbiosis because the symbiont that was brought inside already had experienced primary symbioses. So when symbiosis happens where the symbiont that is, basically an endosymbiosis happens where the symbiont that is brought inside already had primary symbionts, that's called a secondary symbiosis. So after this secondary symbiosis, the model would say that some of the descendants after this secondary symbiosis lost the second mitochondrion. There's probably not much selective pressure to have two distinct mitochondria within a cell that probably carry out the same functions. But the chloroplast is a new thing here. This original host cell couldn't photosynthesize. So getting this chloroplast is really important to it if it wants to use the energy of light. This second nucleus, probably also not essential in many cases, and it could have been thrown away too. In that example I showed you of the cryptomonad, it turns out that some lineages have not gotten rid of the second nucleus. Some have kept it. But then eventually this became this secondary symbiont, a tight affiliation with the host. And now I'm going to show you a model that would explain what happened in one of those lineages that is not in the Plantae group, but has chloroplasts. So Euglenas, if you remember Euglenas, if we look at the nuclear genome of Euglenas, that tells us that Euglenas are related to kinetoplastids, and they are in the excavate group. This is one of the reasons I gave you that big tour of all the eukaryotic lineages. Otherwise, we wouldn't be able to discuss what's going on in any of these lineages. So here are Euglenids. The nucleus tells us what they used to be like. The model to explain this would be that a member of this Euglenid lineage sometime in the past brought inside of it an endosymbiont from the Plantae group. What endosymbiont did it bring in? It turns out we can figure that out by going back to that chloroplast tree I showed you a little bit ago. Now, all chloroplasts are monophyletic. They share a common ancestry to the exclusion of any cyanobacteria or anything else. But if you look within the chloroplast tree, you see some really weird things that are not congruent to the evolution of those organisms once they have the chloroplast. So for example, here's Euglena embedded in the middle of a group that includes the chlorophytes. That's how I've colored them by green. It's sort of the chlorophyte branch. So the Euglena chloroplast is evolutionarily derived from a chloroplast from chlorophytes. So the model to explain this would be that you had a lonely excavate in the lineage leading up to, in the Euglena lineage. It had a nucleus and a mitochondrion. It then engaged in a symbiosis with a chlorophyte, brought that inside of it. We can tell that because its chloroplast is most closely related to chloroplasts from chlorophytes, as opposed to from other plant A members. And this now explains the evolutionary history of all the different parts of the Euglena cell. So there was a secondary symbiosis, which I've drawn by this red line, to explain the evolutionary history of Euglenas, the nucleus, the original nucleus, and then bringing in this chlorophyte, which had a nucleus, a mitochondrion, and a chloroplast. And mostly it's kept the chloroplast, although there are some lines of evidence that suggest that the second nucleus may have some relics left over in Euglenas. Let's do this again with diatoms. Diatoms are not in the Plantae group. We can't explain them by this simple model of chloroplast origin in the common ancestor of plants. Diatoms are in the Stramenopiles, related to brown algae and oomycetes. Here they are over here. To figure out where they got their chloroplast, again, let's look at the chloroplast phylogeny. And here are diatoms embedded within the red algal part of the chloroplast tree. So that tells us that there was a lonely, sad, non-photosynthetic Stramenophile that brought inside of it a red algae via some type of engulfment and then endosymbiosis. And this is what led to the evolution of that, at least that lineage of diatoms where the data came for this chloroplast. So another secondary symbiosis event. Are there any questions about these secondary symbioses before I move on? Basically, what I was saying before the fire alarm was that this is, one thing I want to show you here is how we use models of evolution to then create a scenario that tells us what an evolutionary tree should look like if that model were correct. And then we can go and look at the data and say, okay, the data does or does not support this model. If it doesn't support this model, we have to come up with a new model to explain the evolutionary history. So it turns out there's good evidence for secondary symbiosis in many other eukaryotic lineages. Apicomplexans are particularly interesting. These are the parasitic species, including Plasmodium, the causative agent of malaria. They actually have this apicoplast organelle, which many people for many years thought was either derived from mitochondria or was its own type of organelle. When eventually genes were sequenced from the genome inside this apicoplast, it was discovered to be related to chloroplasts, and the evolutionary history of apicomplexans pretty clearly involved a type of secondary symbiosis. And in apicomplexans, they have then lost photosynthesis in their chloroplast, but they have retained many functions that originally were in cyanobacteria and then in the chloroplast and eventually still there in this non-photosynthetic chloroplast. Turns out that some drugs that target either plants, in particular if they target the chloroplast of plants or target bacteria, can kill apicomplexans because the apicomplexans have some relic processes from their ancient history with a chloroplast. So it's also seen in dinoflagellates and a variety of other lineages. So we have the secondary symbiosis model that explains a lot more of the data. It turns out it's still doesn't explain all of the observations that we have of chloroplasts, chloroplast phylogeny, and eukaryotes that have chloroplasts inside of them. An example is another dinoflagellate that actually appears to have way too many membranes to have undergone just a secondary symbiosis, if you look at this in the microscope. And to explain this organism, as well as a variety of other organisms, including some brown algae, it's necessary to propose a tertiary symbiosis. Here's the model to explain a tertiary symbiosis. Again, you started with a normal eukaryote that did not have photosynthesis. It just had a nucleus and a mitochondrion. And it engaged in a symbiosis with an organism that already had a secondary symbiotic event, like a member of the Euglena lineage. It engulfed that, brought it inside, and now you have an endosymbiosis where if each of these lineages kept all of the different genomes that they had acquired or inherited from their ancestors, you would have one nucleus from this new host and its mitochondrion, a second nucleus and a mitochondrion from the second host, and a nucleus and mitochondria and chloroplast from the algae that was brought into the Euglenid. So now we have 7 genomes, 7 different histories contained within this one lineage. And I haven't talked about it a lot, but one of the reasons that makes evolutionary reconstructions of some organisms very complicated is that the DNA does not stay inside each of these compartments. DNA from the mitochondrial genome moves to the nuclear genome. DNA from the chloroplast genome moves to the nuclear genome. Occasionally it moves to the mitochondrial genome. The DNA from one of these nuclear genomes can move to the other nuclear genome. So even when you lose the chloroplast or lose the mitochondrion or lose one of these other nuclei, some of the DNA is still left behind inside the parts of the compartments that are left. So eukaryotes that have undergone this type of evolution are a bit of a mess when you're trying to infer their evolutionary history. And without this model, you would have no hope of sort of understanding the history of many of these different groups. So there are multiple tertiary symbiotic events that have been proposed, including one with animals. So there's a sea slug that has secondary symbiont containing cells that live inside of it that appear to help it do some types of photosynthesis. There are some suggestions that some of the brown algae have also undergone these tertiary symbiotic events. So it's pretty weird, I get it, but it appears very robust, at least, that there were these secondary symbiotic events, and pretty reasonable that there were, in a few cases, these tertiary symbiotic events. There may be many other symbiotic events that we don't know of, that if an organism has lost its chloroplast, like the apicomplexans have, they've lost the photosynthesis but kept the plastid. If an organism lost the chloroplast entirely, nobody would have even looked for it possibly having history of weird symbiotic events. So it turns out that this general process that I've outlined here with a eukaryotic cell with a flexible membrane that works like an amoeba, bringing inside of it another cell, that's a common process in eukaryotes. That's how many of them feed. That's how white blood cells work. That's how many of the amoeboid organisms work. That's how many of the different lineages of eukaryotes work. Most of the time, they actually digest what they bring inside. But occasionally, what they bring inside doesn't get digested. Eventually, in some cases, the organisms evolve a mutually beneficial arrangement, and then each of those organisms, each of the different compartments get transmitted to subsequent generations. And then we see these weird history of symbiotic events. It turns out, as we will see later in the course, this is a general phenomenon in many eukaryotes. There are hundreds to thousands of other examples of endosymbioses in particular branches of the eukaryotic tree that don't involve chloroplasts and mitochondria. So legumes have nitrogen-fixing bacteria that they bring inside cells in their roots as endosymbionts. Aphids have amino acid synthesizing bacteria that live inside cells in their gut. These giant tube worms in the bottom of the ocean have chemo-autotrophic bacteria that live inside of them. Many other examples we will see throughout the course. There are also many other types of symbiotic events between eukaryotes and microbes, especially bacteria and archaea, but occasionally microbial eukaryotes, that don't involve bringing the organism into the cell. It appears in general that eukaryotes, sort of a preferable or an easy way for eukaryotes to evolve a new function, is to, rather than inventing that function themselves, to acquire the function via symbiosis with another organism. And so what we're going to do now is switch over topics. I'm still going to continue on a topic that originally was going to be in Lecture 9, but I've just added to the slides here that say Lecture 10, which is an alternative. Oh, yeah, question. Yeah, so the question is, have we ever actually seen an endosymbiotic event? So actually, what I study in my lab, or what I've studied for most of the last 10 years, is trying to find recent endosymbiotic events in order to better understand the original interactions between a host and a symbiont. So there actually are gradations of all four types of symbiosis, including what are called episymbionts, that are like bacteria that live on outside of cells of their host. And then there are hundreds of examples of endosymbiotic events that are recent in evolutionary time. Still, most of them that have been studied are millions of years old, but not quite as old as the chloroplast and mitochondrial symbiosis. And then in the last couple of years, some researchers, not me, but other researchers have been trying to, in essence, force endosymbiotic events to happen by making a eukaryotic host deficient in some process and introducing it to a bacteria that has that process and trying to evolve them to some point where they become mutually dependent on each other, and even trying to get them to take them up into the cells. So there are a few examples of forced symbiotic events. And by all of that information, we actually know a lot about how these types of interactions evolve. Does that answer the question? Okay, so we're going to talk. Yeah, another question. I couldn't hear it. Can you repeat it? How would you force it to what? Go out there. Well, you should talk to this guy, Greg Bear, a science fiction writer who's trying to write stories like this. So, bacteria basically won't grow in a new environment unless you provide a selective reason for them to grow in that environment. So I think I was mentioning in one of the sections that people have been trying to inject as tattoos various glowing bacteria and photosynthetic bacteria. They don't like to stick around for a long time unless you have to put in some type of selective pressure in most cases to keep them in those new environments. So people have been trying to enhance humans via symbioses, and so far most of them don't work very well. Okay, so what I want to talk about now is what happens primarily in bacteria and archaea, but also occasionally happens in eukaryotes. It's an alternative to engaging in symbioses to acquire functions, and what bacteria and archaea do a lot of, and occasionally microbial eukaryotes do a little of, is acquire DNA from another organism rather than acquiring a symbiont. Why have to, have an interaction with another organism when you can steal their DNA, basically? And so I'm going to take you through a couple of examples of this and then talk about the implications of it. So just really quickly, antibiotic usage is spreading across the planet. We're putting antibiotics into all of our food. We're putting antibiotics into animal and plant agriculture. We're putting antibiotics on iPhones and kitchen counters and in all sorts of other places. Sometimes that might be useful, but one negative consequences of this is that it selects for antibiotic-resistant versions of particular bacteria that maybe by a mutation in their replication process, they change a gene that allows them to be resistant to a particular antibiotic. That resistance spreads, and those antibiotic-resistant bacteria could, in theory, affect our health. Much more vexing is the issue that when one organism becomes antibiotic-resistant, it's not just that organism that has to infect you, but the DNA from that organism that codes for the mechanism by which that organism becomes resistant to antibiotics can spread to other organisms. This is very common, unfortunately, in bacteria. And this, therefore, you can have the evolution of antibiotic resistance in some organism that doesn't affect human health, for example. But if those organisms get exposed to organisms that do affect human health, those human-associated organisms can become antibiotic resistant by stealing their DNA. Another sort of classic example of, so that stealing of the DNA as lateral gene transfer, we'll come back to what that means in a minute. Another classic example of this involves the bacterium E. coli. So if you take E. coli, normally lives inside of our gut. Most of the strains of E. coli that are known don't harm us. There are a couple of strains that make us very, very sick, very, very unhappy. When researchers read the entire genomic content of different strains of E. coli, they found that those strains shared, an individual E. coli genome has about 5,000 genes. If you compare the different strains to each other, they would share a few thousand genes, certainly, let's say 3,000. Very, very similar to each other. This sort of defined the core of E. coli. If you did the same exercise with different humans, different members of our species, or even compared humans to other primates, what you would find is that we would share 99.5% of our genome with other members of our species, and basically we would share almost all of our genes with even other primates. And when people compared the genomes of different strains of E. coli, this is a Venn diagram showing the genes that they share with each other, even though they share a large number of genes, about 3,000 genes with each other, each of the strains has hundreds to thousands of genes not found at all in the other strains. I mean, there's nothing like this in comparisons of animal evolution or plant evolution. And these unique genes that are found in one strain of E. coli and not others are largely the result of that strain acquiring DNA from a foreign source, and that's why its close relatives don't have that piece of DNA. And in many cases, the genes that are acquired are things like the code for toxins that are produced by the dangerous E. coli, or virulence factors that allow them to invade host cells. So lateral gene transfer is a big deal in bacteria, also in archaea. It's responsible for all sorts of interesting adaptations. It does occur in eukaryotes. It's just not quite as common as far as we can tell. So what I'm going to do now is take you through just a little sort of way to think about lateral gene transfer. And the way to think about it is, I think, to remind yourselves of how bacteria and archaea reproduce much of the time. So they reproduce by this binary fission process where you take a cell, makes a copy of its genome, and then splits in two, and the daughter cells each get a copy of the genome. If you draw the cells, you can draw a cell tree. It's kind of like an evolutionary tree, but it shows the evolutionary history of individual cell divisions. So we start with bacteria, undergoes binary fission to produce two daughter offspring. At the beginning, sometimes it makes perfect copies of itself, and eventually you get lots of offspring of this binary fission process. Some of the time, cells will make a mistake when they copy their genome. That's called a mutation. So this new cell is a little bit different than the parental cell or the sibling. You now have other mutations arising, and you get population variation in your population. Sometimes they will reproduce at different rates, so you can have two divisions here at the same time you had one division there. And lateral gene transfer is what happens when in one lineage DNA moves from, say, this cell to that cell. Now, in terms of this cell tree, it's not that interesting, but if you replace this cell tree with an evolutionary tree of species, and we draw a tree of bacterial species, where this is, say, a cyanobacterium, and these are proteobacteria, and these are spirochetes. It turns out that the proteobacteria can acquire DNA even from another phylum of bacteria. So they can take up DNA across vast evolutionary distances, incorporate it into their genome, and change their genetic properties, and then transmit those traits to their offspring. So it's conceptually similar to the endosymbiotic events we were talking about, where you have two different lineages coming together and then creating a hybrid, which now then its hybrid status is transmitted to future generations. It's different in a few ways, but it's DNA moving instead of an organism joining up with another organism. In sexually reproducing eukaryotes, DNA can merge together by the process of sexual recombination. So you can have two different lineages within a population that can merge together to produce, first haploid gametes, and then those merge to produce a diploid individual, and then it can transmit its traits to subsequent generations. In general, when an event like this happens within a species, where DNA from two different lineages merge together within a species, we're going to call that recombination. So if I go back to this situation, if this was just different lineages within the E. coli species, and DNA moved from one strain of E. coli to another, which is common, that's recombination within the E. coli population. This is recombination within sexually reproducing eukaryotes. In sexually reproducing eukaryotes, this recombination usually happens within species. You can occasionally get hybridization events between closely related species, but you never see these recombination events happening across great evolutionary distances by sexual reproduction. But in bacteria and archaea, DNA can move even from archaea into bacteria. Massive evolutionary distances can be traversed by this lateral gene transfer general process. The book has a little sort of sketch of how to think about in terms of phylogenetic trees the lateral gene transfer process. So if you imagine the core of some lineages evolving by vertical evolution into these four lineages with B and C as sister taxa, next most closely related to D, and then A is the outgroup relative to them. If there was a movement of one gene, let's just say, from species C into species D by lateral gene transfer, if you built an evolutionary tree of that gene, it will look like this, with C and D as sister taxa rather than B and C as sister taxa. So you have, in this case, if that lateral gene transfer happened again, you would have genes would have different phylogenies within one organism, just like the mitochondria has a different phylogeny than the nucleus. Are there any questions about lateral gene transfer before we get into the beginning of extremophiles? So the reason to sort of cover this, one of the reasons is to get you to think about that different parts of a genome can have different histories. We've talked about this with duplication of elongation factors. We've talked about this with organelle evolution, and we've talked about it with lateral gene transfer. So drawing a single tree to represent the evolution of organisms, it just doesn't work. There is no way to do this. Evolution is much more complicated than that. Fortunately, the core of many organism lineages evolves in a tree-like manner much of the time. So after the mitochondrial evolutionary event, the mitochondria and the nucleus co-evolved with each other in a tree-like manner. After the chloroplast evolution event within plants, the plantae group, they co-evolved. It's only these sort of rare events that provide discordance, but there are enough of them that you can't draw a single tree to represent the evolutionary history of everything. Okay, so we're going to start on talking about the biology of extremophiles, and what this is also about is how we study microorganisms. And so we're going to go through a couple of examples of how we study microbes via field observations, via growing them in the lab, and that's called culturing, and then probably won't get to it today, but we'll get to it in the next lectures, what I call CSI microbiology. Just first, we've talked about extremophiles briefly, but we're going to use them as an example today. They're very unusual in one general sense. That is, the definition of extremophile is basically an organism that lives at the end, the tail end of the distribution pattern of organisms on the planet relative to a particular condition. So some organisms grow at very, very high temperatures relative to most other things. They're called thermophiles. They prefer high temperatures. Some grow at high salt, some grow at high pH, some grow at low pH. If they grow at the edge of those conditions, they're called extremophiles. And what we're going to do, in part, is ask how we can go about determining some information about the microbes that are found in particular extremophile environments. And these two general questions are frequently asked when you find a new environment and you want to characterize the microbes that are from there. Who are they? That is, what kind of microbes are there? And what are they doing? What are their functional processes? Again, we're going to apply this to extremophiles, but the principle will work with any type of microbe or any type of organism. So one way to do this is with field observations. So you find a new environment, you think it's really interesting, you can go study the microbes there. You should, from BIS2C and maybe just from generally knowing about biology, be familiar with how people do things like this for plants and animals. You can go out and collect butterflies and study them in the field. You can do transects across a particular environment, sampling along a line. You can use satellite imagery to look at trees and other structures. You can do an organized structure called a quadrat to sample a particular field site. You can use special tools like binoculars. He doesn't know the birds on his head. Or you can go to environments that you wouldn't normally sample using specialized gear to get to those environments and do field biology, field ecology, and do there's lots of interesting stuff in plant and animal world doing field work. The same general approaches can be used for microbes. So we can go out to field sites like a Yellowstone hot spring and collect the microbes that are there and look at them. There are transects studying microbes across global surfaces. We can use satellite imagery to study the products of some or parts of different microbes, like chlorophyll that absorbs at particular wavelengths. We can do quadrats, we can use specialized tools, and we can use specialized equipment to go to environments that we wouldn't normally sample. All the same general approaches apply for microbial field studies that apply to plant and animal field studies, with one unfortunately major caveat. Now, there's great work in this area, but there's one really big caveat, which goes back to this issue of, does the appearance of a particular microbe tell us who it is or what it is doing? And in general, the answer is no. Now, there are examples in particular in certain lineages of microbial eukaryotes where their phenotype is informative to who they are. I talked about this in one of the previous lectures. There are occasionally cases where the phenotype also tells you a lot of detail about what those organisms are doing. But in particular for bacteria and archaea, and for some microbial eukaryotes, this just does not work. And so one thing that microbiologists have done is to say, okay, I can't figure out exactly what's going on in the field. Let's bring organisms back to the lab and study them in the laboratory and try and learn a lot about them in the laboratory and eventually apply that back to understanding what's going on in the field. And one very important tool in doing this is a method called culturing. So the way culturing works is you go out to a particular environment and you try and grow the things from that environment in the laboratory. The general term culturing, sometimes also called cultivation, is just whenever you grow microorganisms in controlled conditions in the laboratory. So you can basically sterilize a thing of milk and see what grows on it. Now, that's not the ideal for studying the biology of individual microbes, because if you just let anything grow on a particular system, there might be dozens to hundreds to thousands of things growing there. And so what many microbiologists try to do is generate what's called a pure culture, and that is growing an individual microbe in isolation from every other organism, and also doing it under defined conditions. So it's a form of culturing. You have to have these controlled or defined conditions, but it's pure as in one organism and not anything else. In almost every case when people do this, those organisms that you're growing are cloning themselves, basically. They're reproducing by binary fission or mitosis if they're eukaryotes, and they're making lots of copies of an original cell. By doing that, you get the organism isolated from other organisms. You can study its biology in isolation, and you can get lots and lots of material by letting it grow a lot in the laboratory and do experiments with it. When you do this, in principle, the way it works is you collect a field sample, you provide specific growth conditions. These may sound familiar to you. You'd like to know something about the energy requirements, where an organism is going to get its electrons and where it's going to get its carbon. You also probably want to know something about other conditions in order to grow different types of organisms, like temperature, salt, oxygen levels, etc. And you go through many iterations until you can isolate a single cell, basically, and get it to make copies of itself. We do this via a bunch of different strategies. You can grow things on a plate of food, or you can grow things in test tubes, in liquid in test tubes, sometimes in sort of a gel material in test tubes. You can grow them in anaerobic chambers where you've removed all the oxygen. There are lots of diverse strategies to getting things in the laboratory and grown. We're not going to talk about all the details of that, but it's really important to get organisms growing in isolation in the lab, because that opens up a huge window of experimental tools to study the biology of those organisms. And so what I'm going to show you now is basically ways that you can go about determining who is out there, or really who was out there when you collected your sample, and what is their potential. Not necessarily what they were doing in the environment, but what are their functional potential that they might be able to do. So who is out there via culturing? It's pretty straightforward. That's basically what Carl Woese did to build the Tree of Life. The organisms that he looked at were grown in the laboratory. Ribosomes were isolated, ribosomal RNA was isolated, sequence, build a tree, and if you have a new organism, you can tell what it is by where it sits in the evolutionary tree. So if you went to an environment and you grew organisms back in the lab, you could have a collection of the different organisms you grew, and for each of them, you could go through this process and assign it to a particular branch in the tree compared to other organisms based upon its ribosomal RNA sequence or other information you could gather in the laboratory. Much more important for studying microbes is to figure out not just who they are from an environment, but what are their functional processes and what are the sort of the potential that they could be doing in the environment. And I'm going to take you through, in the last few minutes here, an example of this for an extremophile. So if you go to a salty environment and you're interested in who is out there in this salty environment, and you take a sample back to the laboratory, let's say you were able to grow some of those organisms in pure culture in the laboratory, you typed their ribosomal RNA, it told you that they were from a particular branch of Archaea, one of the Eukaryotic, this group called the extreme Halophiles. Those are the organisms that like high salt. When people first started doing this, they didn't know that that group of organisms would be called extreme Halophiles, so I'm going to show you how they figured that out. So basically, if you go to an environment, you grow a particular type in the lab, you take a single clone of that organism, let it grow for a little bit to make some copies of itself, and then you split that sample into multiple distinct, say, test tubes, and you provide it with diverse conditions, and then you monitor the growth in those different conditions of those genetically identical samples. And I'm going to show this to you visually rather than with words. So you go to a salty environment, you grow something in the laboratory, you grow what's called a starter culture, you take a sample out of that and you put a little bit in these different tubes. First, you set up those tubes to have different growth conditions. And then you let the organisms grow. Let's say you were interested in salt, how the organism grew in different salt concentrations. So the different conditions, you could use 1 molar salt in one of them, 2 molar, 3 molar, 4 molar, everything else being exactly equal. One of the reasons defined conditions are really important. You're now comparing genetically identical strains where there's only one difference between their growth conditions, the amount of salt in those conditions. You monitor the growth over time. You can look in these test tubes at how dense the culture is by basically how much light it absorbs or how much light it reflects if they have a color reflection. Monitor it after 1 hour, and you can see it's getting, this is the, oops, shouldn't go backwards. Monitor it after 1 hour, 2 hours, 3 hours, and you can start to see where the growth is occurring at the greatest rate. You can plot this growth over time. You can convert that plot of growth versus time to the rate of growth for each individual condition and get a plot like this. This particular strain grew best at 3 molar salt concentration. And then you can do the same thing for strain number 2, strain number 3, strain number 4. You can do that for strains from different salt ponds. You can do that for strains from different environments. And then you can ask, what are the preferences for different organisms as to what salt concentrations they like to grow at? And you can make a plot like this, salt concentration on the x-axis, growth rate, like by the method I showed you there, on the y-axis. And then different types of organisms have different patterns here. So E. coli hates salt. If you give it just a tiny bit of salt, its growth rate starts to plummet. But other bacteria, so that's called a non-halophile. Some organisms prefer to grow at low salt concentrations, but will tolerate higher salt better. Those are called halotolerant. For the organisms that actually prefer to grow at high salt concentrations, those are called halophiles. They prefer high salt. And the halophilic archaea are way off over here on the right, preferring to grow at incredibly high salt concentrations. And what you can then do, once you have this list of organisms that grow at different salt concentrations, once you have them isolated in the laboratory, then you can do experiments that compare the halophiles with the extreme halophiles, with the halotolerant organisms, with the non-halophiles, and ask, what allows them to grow under these particular conditions? And for example, in general, if you take a cell and you put it into an environment where there's lots of salt in its environment, that's what I'm trying to draw here, salt in the environment, here's the cell. The water is going to go out of the cell, it's going to get desiccated by osmosis. The water sort of prefers to go to where there are higher concentrations of these salts. So since the water prefers to go where there are higher concentrations of things dissolved in these conditions, one adaptation that has been found in many organisms that grow in high salt conditions is to fill up the inside of the cell with what are called compatible solutes, other compounds that compensate for the fact that it's living in an environment where there's lots of stuff dissolved in the water. And that keeps the water from going out of the cell. Most of the organisms that have been looked at that either are halotolerant or halophilic fill up the inside of the cell with things that you might expect, with proteins and carbohydrates that they're already basically making and doesn't really change a lot of the functions on the inside of the cell. The halophilic archaea, these extreme halophiles, do something very weird. They fill up the inside of the cell with other salts. So they're inside of their cell, where their DNA is, where their ribosomes are, where their enzymatic processes are happening, is all occurring in basically a hypersaturated salt solution. And this is interesting for many biological reasons. This is just to point out that the halophilic archaea are monophyletic. So this evolutionary adaptation happened once. Oh, and I must have taken out the other slide. One of the reasons that this is really interesting is that there are many human industrial and agricultural processes that we would like to have occur in high salt solutions. Many people who work on these industries have been trying to find enzymes to add to these reactions that work under those high salt conditions. A great source for such enzymes are these organisms that fill up the inside of their cell with salt, because all of their enzymes already work at high salt concentrations. And we will continue a tiny bit on extremophiles in the next lecture and then get into the human microbiome.